Let's move on, and now I'd like to pass on the floor to the representative of Vector Best, which is the official sponsor of the conference, and that is Natalia Fomenko. She's gonna talk to us about the epidemiology study of poultry infections. Vector Best organized a field trip for us for tomorrow to the poultry farms that we were unable to visit the day before yesterday. Thank you for giving me the floor. It's late in the evening. Everyone is tired, so I'll be brief. But fast. This is the epidemiology study of poultry infections. But I will just share my views and my take on things that we see when we try to diagnose, run a diagnosis based on a comprehensive approach. And it's not about diagnosing a single bird, but rather an entire poultry farm. The very last slide of my presentation will give you a great example of this particular statement, that there are lots of diseases and you might not know what the dominant pathogen is, and oftentimes what we see is as follows. When people turn to us for help, they seem to know what pathogen circulates in their form, but their knowledge is only limited to 10%, and they don't know 90% of the pathogens circulating there. They don't know what the dominating disease is. And that means that we need to employ comprehensive approach when we run diagnosis at the level of a farm. We collaborate with Mr. Leonov from the Veterinary and Agro-Industry R&D Center in the Russian Forest. We try to track the link between the identified pathogens. We used samples from spleen, duodenum, ileocecal glands, trachea, and lungs. And we analyzed over 800 samples from 3 different federal districts and 5 different farms. We did screening for quite a wide range of various pathogens, those DNA and RNA containing, for instance, mycoplasma, if we identified it, infectious bronchitis, and identified highly pathogen Newcastle subtype. And we did sequencing if we're unable to run PCR tests. Over the years of collaborating with vets and zootechnicians, we came up with a number of rules that are applied to all the diagnosis sets that we used. A highly positive sample was the one containing up to 30 CTs. If you do PCR tests, you know what I'm talking about. That means that the pathogen concentration is so high that when you see figures like that, you really understand that this is the clinical stage, when the clinical signs are visible. 30 to 38 CT, if we're talking about ELISA assay, that's a gray zone. It's either the start of the infection stage, or it might mean that infection has been curbed by the vaccines, and this is just the remains of the pathogen. PCR can be used both with and without antibodies after vaccination. So what we have seen so far is as follows. This is the summary of all our sample tests. IBV is the virus that isolated in almost 50% of samples. 20% is the share of high burden IBV. Circovirus and the reovirus followed IBV. Identifying the type of IBV, the ND and mycoplasmosis. IBV, well, 793B was the strain that we isolated most frequently. But we haven't seen a D274 strain at all. The Mycoplasma synovia was the one that we isolated in the samples, and the low pathogenic ND was also isolated in the samples that we analyzed. As for breakdown of IBV strains by poultry farms, what we see is that they have each farm had its own range of strains. Farm 1, IBV breakdown. Despite the fact that all three strains were isolated, nevertheless, the QX strain dominated. It accounted for 35%. As for farms 3 and 4, the situation looked similar, but I mean, all three strains were isolated, all three subtypes were isolated, but for farm 3, we see the fact that the mass strain is still present, and despite the fact that other variants were also isolated. As for farm 4, we almost did not see any mass strain. That's as far as bronchitis is concerned. Now let's take a look at the broiler. At the broiler farms. Farm 1 is the most successful out of the 4 presented here. As you can see, the columns are almost of the same height, and there's almost no difference. They did their best, and they were almost on the verge of dealing with all the diseases. На первом предприятии были проблемы с санитарной обработкой. Там не домывали, и поэтому вот инфекционные агенты, они кочевали каждый раз и оставались... Farm 1 ran into problems with cleaning and washing, and that's why these pathogens remained remained in hen houses and affected. Beetling bugs were found there in huge numbers, and we identified all the pathogens in these bugs. As for Farm 3, the IBV was an obviously dominating disease. Reovirus and psychovirus dominated in Farm 4. Days 15 and days 30 were the peaks in mortality in Farm 4, and this farm produced carcasses with the lowest quality. That was brought to my attention, and when the pathogen breakdown looks like that, uh, it affects the quality of the carcass. The bacterial pathogens affected the digestibility and the absorption. Farm 5 looks similar to farm 1 because, you know, the columns are not as high, the distribution is quite even. However, what we see is the starting point of the problems because the circovirus and the Gumboro disease were at the beginning of the outbreak, actually the farm ran into huge problems and they had to undertake emergency measures to deal with the outbreak of all diseases. As for the isolation of pathogens broken down by days, so the situation looks like this. Newcastle, IBV, and Mycoplasma are shown on the left hand upper diagram. Until a certain point of time, IBV and NV are going down. I mean, you vaccinate the flock, the situation improves, but then both curves go up. And mycoplasma, that used to be almost at zero, goes up as well. The second diagram shows data for AIL, laryngotracheitis, and its response to the Newcastle disease vaccination. And the second peak was caused by the response to the Gumboro peak. Gumboro and circovirus remained at a high level while the AIL went down. And I just wanted to focus your attention on IBV, infectious bronchitis, that was in the lead and created most of the problems for this specific form. In the left-hand side bottom corner, you can see that the IBV accounts for the largest share. The right-hand bottom corner shows the breakdown by subtypes or strains of the IBV. MES strain accounts for almost 30%, QX for almost 50%, and 793 B for 3/4. As for identification of these subtypes broken down by days, shown on the left-hand side are the samples that with positive response, and shown on the right-hand side are samples with a high burden. The vaccine worked perfectly well. As you can see, from 60 it went down to almost zero as far as the mass strain is concerned. You no longer isolate mass strain. What happens with QX? The QX is detected, is isolated more and more frequently, but by day 31, the 793B also started climbing up. We stopped at day 31 because it didn't make any sense to sample anything because all the birds died. That's the fourth farm. At the bottom of the screen, you'll see the diagram showing that circovirus and reovirus were the main two problems. However, bronchitis, though not in the lead, was also there. Both circovirus and reovirus and the Gumboro disease curves go up, and you know, the figures are quite high. I'm almost done. At least here, everyone survived until day 42. For IBV, it's basically the same image. The mass strain was stamped out with the help of vaccine, while the other two modifications did not respond to vaccine. And this is a layer form. We see some infections that were controlled well. The laryngotracheitis was not a problem there. However, uh, avian influenza was a problem, mycoplasma was a problem, and chikungunya virus was also a problem. So hemophilus, there was a breakout of outbreak of hemophilus that resulted in a mortality of 30% of non-vaccinated layers. That's just a comparison of samples from ileocecal glands and duodenum. In ileocecal tissue, the isolation frequency is higher, but I like to deal with intestines. That's the best biomaterial samples ever. And this is the last slide of my presentation, proving the statement that I showed you on slide 1. This is data coming from abroad. Biomaterial was sent to us, and they said that the problem was Newcastle disease genotype 7. They couldn't solve the problem. So we analyzed it for ND genotype 7. It is it, indeed it's there, but what we saw was the bronchitis and the reovirus and the Gumboro disease. And they did their utmost. They spared no effort to deal with the Newcastle, but they missed out on all the other diseases. Anyway, PCR allows you to monitor all the pathogens and you can rank the various pathogens depending on their importance so that you understand what to focus on. Thank you so much for your attention. Thank you. Now it's time for the Q&A. Just a clarification. Do you differentiate between vaccine strains and wild strains, field strains? Because, you used live vaccine, right? No, we didn't differentiate. We only used mass strain vaccine. So you didn't. Right. Thank you so much, Natalia.